Hi, welcome to the Network Analysis in Systems Biology course. My name is Avi and I'm the course director of this course. This is the second iteration of this course. This course was created by the entire Mayan lab, not just by myself, and you're going to meet the members of the lab throughout the course. We are a research dry lab in New York City at the Icahn School of Medicine at Mount Sinai. This version of the course has many new lectures, as well as some of the lectures from the first iteration of the course. We also removed some of those lectures from the first round of the course, and all of those lectures are available on my YouTube channel if you're interested. In this lecture, I'm going to give you an overview of the course. So the first module is just the introduction, including this lecture. So in the second module, I first describe some methods of data normalization and data cleaning, which are prerequisite before we start data analysis. Then this module is dedicated to a new method that we developed in the lab called the characteristic direction. And this method, the characteristic direction, significantly improves on all popular methods for identifying differentially expressed genes from microarray or RNA-seq studies. Neil Clark will describe in 4 lectures all the inner working of this method, and then how we benchmark this method against the existing popular methods to identify differentially expressed genes. The next module will cover gene set enrichment analysis and several tools that we developed in the lab that are related to gene set enrichment analysis. These tools are called Enricher, Expression to Kinases, and Drug Per Seeker. Enricher is a gene set enrichment analysis tool, and it has probably the largest collection of gene set libraries. We now have added to Enricher many new gene set libraries, and we have almost 60 different gene set libraries in many different categories. As you can see, the tool is becoming popular, and you can see that already over 96,000 gene lists have been uploaded. Expression to Kinase is another enrichment analysis tool that identifies in an alternative way cell signaling pathways upstream of differentially expressed genes. So basically, expression to kinases identifies the transcription factors upstream of differentially expressed genes using enrichment analysis, and then those transcription factors are connected through known protein-protein interactions, and then this subnetwork that is connecting the transcription factor is inputted to a kinase enrichment analysis tool called KIA to identify the kinase, the protein kinases that are most likely phosphorylating many of the substrates within this subnetwork, potentially regulating the differential expression. The third tool, Drug Per Seeker, used data from the old as well as the new connectivity map dataset that was developed by the Broadin to identify small molecules as well as approved drugs to either reverse or aggravate gene expression given signatures of up and down regulated genes in situations such as disease or other experiments that measure genome-wide gene expression under 2 conditions. Next, we will cover processing of deep sequencing datasets, including RNA-seq and ChIP-seq. The same lectures that were presented in the first iteration of the course by Yan Ko remain, as well as a new lecture that describes a platform called STAR that is an improved aligner for RNA-seq processing. The following module describes various methods for performing unsupervised clustering. The lecture will describe principal component analysis, hierarchical clustering, self-organizing maps, network-based clustering, and show various applications where those methods can be applied to. The next module will describe many of the resources we processed for constructing the gene set libraries for Enricher. These resources are online, open, available, structured datasets that can potentially be integrated into a global framework that can connect genome-wide experiments that were collected from mammalian cells to mammalian phenotypes, including disease, mouse phenotypes, and side effects. Here is a list of the various categories of resources that can be converted into gene set libraries, networks, and bipartite graph. And this is a description of a network that show how all those resources can be bound together to form a global puzzle that can connect genotype to phenotype. And if you're interested in more details about how this can be done, described in a recent article that we published that is available as part of the supplementary materials for this video lecture. The final module, like the last time, is about crowdsourcing. For this iteration of the course, we designed 3 crowdsourcing challenges that we hope you will be willing to participate in. The first crowdsourcing challenge is a microtask, and the idea is that together we're going to build a gene set library from the GEO database by collecting studies that knock out or knock down or overexpress a single gene in mammalian cells. You are asked to identify such studies and post them into the crowdsourcing website. The other 2 crowdsourcing projects are megatasks. Those are hard challenges. The first one will provide you with gene expression data after drug perturbation from over 400 drugs, and also a matrix that describes side effects that are known to be associated with those drugs. The goal of this megatask is to develop a classifier that can predict side effects from the gene expression data. If this works, such classifier can be used to predict side effects for drugs that are not yet marketed. The second megatask will provide you with a large set of gene sets without specifying where they came from. Your challenge is to identify the structure within this gene set and provide detailed analysis that describe the dimensionality of all of those gene lists. For the first microtask, we developed a Chrome extension called GEO to Enricher. This Chrome extension adds buttons to the GEO database, so we are excited to have you test this new tool. So again, thank you so much for signing up for the course and watching those videos. We hope that you'll find them helpful for your research and enjoy the overall experience.